let's start today's journal club. Hello, I'm Timo, and today I will talk about dimensional reduction method inspired real biological circuit. And at first, let's read the abstract. Read. So, where is the gap sentence? I think this is the especially at the latter part. The implementation in simple biological circuits remains unclear. So, our sensory input is high-dimensional data. For example, in olfaction, it means smell. Number of types of olfactory receptor in human is around 400 neurons. So, if this chemical enters in our nose, 400 olfactory receptor responds to these chemicals. So it means that response is high-dimensional data. So yeah, high-dimensional data is difficult to interpret. So we can understand what smells. So in order to interpret the smell easily, our brain may reduce dimensionality. If the high-dimensional data is mapped to simple low-dimensional data, we can easily understand this chemical smells sweet. One of the simplest dimensionality reduction method is principal component analysis. And this method is widely accepted. But there is a problem. PCM method cannot separate entangled data. So for example, if the input data is entangled ring, low dimensionality reduction data also entangled. So we can distinguish the blue ring and red ring. So our brain need to separate in a nonlinear way. And one of the nonlinear methods is neural network. The neural network approach can separate entangled data. It's very good. But it is unknown whether real biological system use the similar algorithm. So the research question of this paper is, can we develop a simple and effective dimensionality reduction method mimicking biological systems? Before that, I will briefly introduce a conventional method. It's called TSNE, T-distributed stochastic never-ending method. We suppose input data is X and output data is Y. X is high dimensional data and Y is low dimensional data. And we compare the similarity between input and output. For example, the similarity between input data points is defined as Gaussian distribution. This graph is distance between two data points. So that this method assumes that this distance between two data points follows Gaussian distribution. In contrast, output a distance between two output data points follows t-distribution because the t-distribution has fat a long tail, so this assumption can avoid crowding problem. Gaussian distribution is just rapidly decay, so and the data point can maybe crowded, but t-distribution avoid crowding problem. And then to minimize Kalbach divergence between input similarity and output similarity, the low dimensional output data Y are constructed by using back propagation. So, I don't fully understand the details, so if you want to know the conventional method, let's read this Nobel Prize paper. This method is very successful and widely accepted, but it is unknown whether real biological system uses this algorithm. Then, the author developed a new method for dimensionality reduction inspired real biological circuit. The important point is this new method is called Hebbian TSA consists of three-layer network. Input network and input layer and middle layer and output layer. These layers correspond to input layer corresponds to projection neuron in Drosophila olfactory system, and the middle layer corresponds canyon cells, and output layer corresponds mushroom body output neurons. So this structure mimics real biological circuit. And the assumption is the dimension of middle layer is higher than input. Actually, the number of canyon cells is much larger than projection neurons. And the output is low dimensional. In this paper, the output dimension is fixed to 2 for visualization, but it's not a limitation. It's the first assumption. The second assumption is projection from Z to Y is linear and adaptive. So Y is called WZ. W is weight matrix. And the weight matrix W is updated to minimize some cost function. Yeah, I will explain later. And projection from X to Z is random and fixed. So the weight of this connection does not change. It's fixed.
But the distribution is random. Distribution of the weight and fixed. What is the meaning?
Yeah, it's the distance. I think they didn't mention the distribution.
Just randomly chosen, and it does not change during the training.
Yeah.
They only train the value of the W.
Yeah. Only trained this connection.
Is there any rationale to set this system? It looks a little bit strange. Architecture.
You might think it's strange, but the author mentioned this architecture exists in Drosophila olfactory system.
Random and fixed weight.
Yeah. Random and fixed. Yeah, it's there is evidence.
And also, I think it really similar to the something called reservoir computing. Yeah. So it's in the reservoir system, they just fix random value there, and they only train the output layer. So maybe in that sense, it looks very similar to reservoir computing.
Maybe I'm not the expert on reservoir computing, but maybe the author knows reservoir computing, and they are also inspired in that. But I'm not sure. Thank you.
The reservoir computing is about the recurrent network. So it generates different types of. It generates basically all the units generate the random patterns, different random patterns, and so you're right that the weight matrix in the recurrent network is fixed. So they just generate the random patterns, and by when the learning comes in, you have to read the local patterns you synthesize them into a certain pattern that we want to express. But this is a feedforward network. So.
Yeah, it sounds similar. Do you have any other questions or comments? Maybe this is a highlight of today. It's okay. And
So is there any mathematical advantage to use the larger dimension for Z compared to X?
yeah, it's maybe it's next slide.
Oh, okay.
Yeah, introducing the middle layer, the update rule of weight matrix is feed forward. It's not back propagation. So we can more easily update a weight matrix than a conventional method. So I think this is the advantage of this structure. And there is also evidence of a feed-forward network. The global factor is the global factor corresponds dopaminergic neurons. Using this structure, the authors developed a dimensionality reduction method. And the details mathematics is here. If similarity is defined as Gaussian distribution between neighbor time steps. A conventional method is not limited time steps, but for this new method is limited, and the distance is between neighbor time steps. And AK of T is looks strange. AK of T is defined by winner-take-all mechanisms. If this K of T is the largest among the middle layer neurons, AK is 1. Otherwise, AK is 0. So, for example, this 1 is 1, this 2 is 5, and this 3 is 3, and this 4 is 1, and 5 is 3. The largest value is 5, this is 2. So only this 1 and 2 is 1, and other AK becomes 0. It calls a winner-take-all mechanism. And the reason that they introduced this mechanism is the winner-take-all mechanism is required for the middle layer to exhibit linearly independent responses. To do so, they introduced the mechanism. And you might think this mechanism is strange again. But they said the mechanism can biologically be implemented by firing threshold. The neuron has some threshold, by using this mechanism can achieve.
Sorry, but what is the meaning of the XK diff?
Okay.
The XK diff, the
XK diff is Gaussian distribution, and denominator is normalization factor. It's just normalization, and the numerator is Gaussian distribution.
What is the meaning of t here?
T is time.
So, do we consider the data which have just time series or?
The input. I didn't mention, but and the times the input data. The data inputs randomly and repeatedly. So it's like times streaming data.
Okay, streaming data, but
yeah.
it's randomly set.
Yeah, random and repeat.
Okay. Is there any reason for
training?
Yeah, for training. Yeah.
And do you have any biological interpretation for winner-takes-all mechanism?
Winner-take-all mechanism is interpreted by firing threshold.
Firing a single neuron, or what is the meaning of the index K?
Index K is the
Each
number of
neuron?
neuron. Yeah.
So that means winner-takes-all mechanism is corresponding to the firing a single.
Yeah. Firing a single neuron. Firing a single neuron, and the other neuron doesn't fire.
Doesn't fire.
So this is just for the training part, so it's linear and then goes to y, right?
Yeah.
We're just talking about blue stuff up there.
It's training of this part.
This XK diff bar, is it time average?
XK diff varies. It's time average. It's normalization.
Yeah, it's a normalization factor,
Yeah.
but it's not time dependent. It's got a bar.
I think it can depend on time.
And anyway, it's only one of these is non-zero at any given time, so then why would you normalize H?
Maybe for simply
It's gotta be a time.
simply calculation. I think there isn't normalization factor, and the algorithm will work. But for simplicity, they introduce the normalization.
So in this so the subscript K means the size of D, right?
Size of
K
input. Size of
The size of D, because AK is determined by DK.
size of Z is S.
S. Then what is K?
One of the sorry. Yeah. The middle layer is this Nemo 1, 2, 3. the size of middle union is S,
S.
and the K is yes, yeah,
Index from 1 to
Index.
S.
yeah, index is
Okay.
is index.
Now, how can you determine sigma K?
Sigma K is sigma K is I didn't explain, but K determined
Common
that
index.
to minimize perplexity. Perplexity means perplexity is defined as this. And this part is entropy. Entropy. So the complexity means a kind of predictability of
Yeah, but then again, what is P? What distribution that P represents?
Sorry, it's conventional method. This is the definition of perplexity. To minimize this matrix, and the sigma is determined.
So, what is the length of A? So A is the coefficient for the input or input. Then the dimension of A is equal to input dimension.
The dimension of A is the same to Z. Yeah.
How about the top the second equation? What is the meaning of the A? Okay, so X have no index.
구조가 이유를 모르겠어, 솔직히.
헷갈린다.
I understand the coefficient AK2 is related to the element of rate matrix.
Yeah. I see. Yes, zero.
Linearly independent responses.
Yeah. That's right. It's 0, 0, 1, 1, 1, 0, 1, 0, 0. Yeah. I didn't really notice that, but that's right. So EK corresponds to elemental vector.
So what is the dimension of the input similarity then?
Dimension ring. The dimension of input is less than that of middle layer.
They use same K?
Yeah. AK is same K. It's, the dimension is the same to middle layer.
Middle layer.
So I guess I'm calling the input X and not the lower layer, which is lower dimensional. But didn't you already say the first layer was, I can't remember what the dimension, 50, and then Y was gonna be like 32? It's like we're overusing X here, maybe? Yeah.
Dimension is,
It says input layer X is 50 neurons.
that's
This is 2000.
yeah,
But then it seems like you're using X as the input
Mm.
from the input layer into the middle layer.
but it's the 50 neuron is real biology.
No.
The calculation
Look.
is
You're indexing by K.
yeah.
You have an AK in the X's. X is 50 dimensional. Z is 2000 dimensional. Right? That doesn't line up. Go forward to the next. There's R and S, right? R and S. Like, how can you have XK lined up with AK? They don't even have the same dimensions.
Yes, I think it's same dimension. Same dimension to zero.
No, they're not. X is 32 dimensions, Z is 2000.
That's it.
R and S. R is greater than S,
Yeah.
right?
Yeah. S is larger than
So Z is larger than X.
Yeah, but they were. R was the dimension of
Z
Z.
layer is larger than X.
yeah. Z is larger than X.
And his question is?
What does it mean, the correspondence between XK diff
Yeah.
and AK?
Yeah.
Well, I guess you're doing a norm difference between the x's, so you're taking the whole vector.
Yeah.
Okay, that makes sense then.
This corresponds to the distribution response Z. The Z response to X.
And so the norm here is like is it the 2 norm? Is it? So it doesn't matter the dimension difference because you're taking the norm of xt minus 1 squared. Right, so then that reduces the entire. So it's, the notation's a little bit confusing because you say x diff k. You expect x diff to have the same dimensions as x, but the definition doesn't say that.
Xt minus xt minus 1 is Euclidean
Yeah.
distance. So it's
2 norm then.
Just norm. And but AK is vector. A has a dimension. So the xk is the same dimension to
Takes a minute to digest it, I guess.
Any other questions?
So here, xk, k index size is same with what?
Okay, the k index and the size of k index is equal to z.
Okay. That sigma k is null or that's also parameter?
Yes, sigma k is various. Sigma k is determined to minimize this index. This part is entropy. So the perplexity corresponds to a kind of predictability.
Predictability or
Yeah.
you predict what?
If the entropy is high, so the prediction, the perplexity is if the entropy is high, the perplexity becomes low. So the predictability becomes low. And if the entropy is low, and the perplexity becomes high.
That seems pretty complicated.
Yeah.
I don't know, so that means according to this, only A2 will be the one, right?
Yeah, only.
That means only X2 will be the non-zero.
Yeah.
So then so then 0 something 0 0 0 0 0.
Yeah.
왜하지?
I think I read with the paper. I think something is different from the like
Can you
but the paper is really confusing. So we will give x1 y1 x2 x r1. This is one data point, and we can give n data points, right? So these are input, and
N is the time? Then
number of input data.
number of the
Okay, we can give n input data, and these are all vectors.
and where is time variable?
Oh, that is not. So they will give this serial, sequential stream of data.
So,
First give x1 and x2.
so here then large N is time index.
Oh, yeah.
Okay.
So then, so for each x1, can we see that?
Yeah.
Oh yeah. We know that this connection is fixed, right?
Yeah.
So we can calculate the value when each xi is given, right? K.
Okay.
So, and this is transformed to 1 or 0, depending on if zk xi is larger than other all zk, for example,
That's
x1,
winner takes all.
and otherwise 0.
Mm.
Then it makes sense.
X가 그러니까 저기 위너 텍스 올로 들어가는 거예요?
네네. 여기서 이놈 위너 텍스 올린다.
그거가 오케이. 그 위너 텍스 올이 나왔고, 그러니까 그게 지금 AK인 거잖아. A.
그래서 여기서 그 zk transform to the ak.
그럼 여기까지는 이해가 됐어. 그러면 지금 AK가 여기 지금 역할을 하는 게 시그마 K를 바꿔주는 것밖에 없거든요. 뒤에 가보면. 그러니까 그러면 저 디스턴스에 웨이트를 얼마나 줄 거냐에 관여를 하고 있는 건데 근데 그 시그마 K가 이걸로 정리된 거가 그러면 의미가 있어야 되는 거잖아요. 근데 그걸 이해 잘 못하겠어요. 그래서
일단 이렇게 하면 뭔가 시그마가 자체 의미는 일단 마련되니까.
okay, so this determining AK means that so anyway the weight is the upper part the front part weight is fixed.
Yeah.
So AK is anyway randomly determined.
Yeah.
And then we only give the winner we takes the winner takes all, then that means only one sigma K of X to define the X div. So that means that sigma K plays an important role in here, but the sigma K is determined in this. But still I cannot understand what is the meaning of maximizing or minimizing.
Thank you, Calvin. That's right. And I also don't fully understand, but it means it corresponds, and this part is entropy. So some ambiguity of the distribution, and the perplexity is negatively correlated entropy. So it's minimum perplexity correspond to maximum entropy. So it means to distribution becomes wide. As the sigma K is determined.
Okay, so if we solve that equation, we sum all sigma with K, right?
Yeah, K. K is correspond. And
Isn't the same as just X hat diff?
Yes.
Why do we need a K there?
Same definition.
Same definition?
XK is and this XK. This in and then this.
Here, K. Do we add this for all T or all K? What is the sigma
Okay.
meaning?
K.
K, then perplexity is defined for each T.
Perplexity depends on time.
Okay.
Sigma depends on time then?
Yeah, sigma. Yeah, I think sigma is also depends on time. The author said it's simple. It's not simple.
So what I thought is that if we want to define what's that? Entropy, we need a distribution. But for each time t, we have one value for each x hat, right? Is it right?
Yeah.
Only one value.
이런 박사 잘 모르겠나?
perplexity, I think perplexity, 아니 이거 되게 단편으로 the perplexity can go back to the degree of aggregation of the points.
Yeah.
in my game.
Yeah.
So if you set some complexity level, then the final point distribution could be changed, right?
Yeah.
So,
Yeah.
그 정도.
Okay. What about I think let's go to the final stage of this algorithm, next page. Come back. Maybe if we look at the goal of this algorithm, we may get some clue. Okay. Anyway, currently we make the so-called X differences. X difference is nothing but the just difference between the distance between two vectors at two different time points. But we give that value at a certain position in the G, right? That's currently what we do, right? And we don't know why still. Is it clear? So this K index will be size of the Z. Right? Am I correct? Yes. And then so x1 will be 0, x2 will be something, and then 0, 0, 0, 0. We will have that value for every t. So for every t, we have a g size vector, g size vector, g size vector. But in this g size vector, at the only one position, we have some value. Still, I don't know why. So basically, we have a matrix. Row is z size. Column is t, and then in this matrix, everything is 0. Some value only appear. I have really no idea why we do this. And that's maybe we call these two arrows are that, right? This is difference is value, and A will determine where. Does it make sense? We have a matrix. So these two arrows are going here, D. Mystery, yeah, we all agree. Let's move on. What's going on?
Similarities. You define this is simple. It's
Okay.
T distribution.
Yeah, we understand one, we understand that.
Yeah. And the weight matrix is update to minimize this global factor. And global factor is defined as x difference minus y difference. Similarity between x distribution and y distribution.
So this is matrix, right?
Yeah.
With the head, we change it to something else.
Yeah, this is not matrix.
X difference.
X differences, and then head is
It should be single value, right?
A single value.
for each time.
For each time. Single value for each time.
What? Then why we made a matrix? Because why I'm confused is across all this K, this will be only one time
Each time
Tested,
only
right?
one control.
and then what is this?
Don't, as far, I don't know, it's just some, are you normalizing the exponential, like a normalized
What is this?
Gaussian?
What is this bar?
It's like 1 over the square root of
Yeah, normalized.
2.
뭐 2파이 루트 2파이 시그마 뭐 그런 걸로 나와야지 한다.
So then it might depend on sigma k, then it matters.
AK가 사라져.
그러니까 AK가 그러니까 진짜 여기서 AK가 역할을 하는 거는 시그마 K를 뭘 쓸 거냐 정해주는 것밖에 없어요 지금.
아 있다고.
네, 그러니까 시그마 K 1부터 D의 개수만큼이 있는데 시그마 K가 그 시그마 K 중 몇 번째 걸 쓸 거냐 이걸 결정하는 것밖에 없어요 지금.
그리고 그냥 원래 자리로 가는 거지.
그게 그냥 x diff 햇에 들어가는 거예요. 거기에 결정이 돼가지고 디퍼런스를 계산한 게 x 햇에 들어가요.
왜 그러지?
So if I understand correctly, this one is value, right?
value, just value. single value.
Each time we have a single value. That value is just this differences, but we
Give some weight.
give us some weight for that difference. How much this weight is depending on this k index.
Yes.
So x difference hat is nothing but just difference for each time,
Each time.
but we give a different weight of
Right.
ak.
Yeah, exactly.
That's all.
Yeah.
Why they make it such a complex thing?
Yeah.
What?
저 A를 왜 저렇게 정했는지 잘 모르겠어요.
불러오면 쓰면
근데 저게 인풋에 따라서 G값이 바뀔 테니까 막 인풋에 따라서 뭘 쓸 건지가 이제 결정이 되는 건데 그것도 랜덤으로 결정된 거란 말이죠.
랜덤.
그럼 저게 정의가 진짜 중요해졌는데.
Like I would rather them just say it was like a max over k or something. And then the normalization in the definition.
So then xk hat difference, that is a final input. This one is
Yeah.
just time series data, right?
Yes.
One time series vector, but each component is difference between
Different.
current time and
Previous time.
previous time. So we give it some weight.
Yes.
Some weight. It's
Unclear.
unclear, but anyway, xk hat is just time series data. Let's forget about the sigma k
Okay.
at this point. Just distance. Good? And then can we move next? And then y diff is
Similar. What?
similar. Y diff t is also distance, but now distance is now in
T distribution.
okay bottom. Sorry. And then y diff.
So sorry, it's
y.
it's y.
Okay. And again, they do some AK thing here as well. No, we don't need it. Why there is sigma K thing? We don't have K index here.
Sorry, it's not sigma. Sorry,
Oh,
it's completely
so
sigma and sigma
I think-
is no.
Yeah,
here we
that-
need a
yeah.
hat, isn't it?
Yeah.
Here,
Maybe some
Yeah.
factor there. Maybe.
just hat here.
Yeah.
This is not the vector,
Yes.
just value. So we need
Yes.
hat here.
Yeah.
Correct.
Yeah, no, it's divided by by different.
Anyway, it's some normalized value.
Anyway.
Anyway.
This one is divided by some value. Anyway, normalize it, it's okay. Anyway, y difference, y hat is just time series difference between yt and yt minus
Okay.
1. Happy? So x hat is a distance difference is time series. Y hat is y time series difference time
Okay.
series. Happy?
Okay.
Let's move on. If we understand this, you will be very happy tonight. Yeah, if we don't understand, you cannot go home. Do your best. Okay? So we have a time series here. And it's the same size? Yes, same size because time domain. So we can subtract it. But to be frank, I don't know why we divide by large n.
What is large n? Large n is time series size?
Yeah. I don't understand this.
Dimension? No.
Is it dimension size?
And
Maybe because the large n appear here.
Oh, because you're summing.
Yeah, summing.
Yeah, but only one is contributing, so why would it scale with n?
Maybe what if the y is in the two-dimension space, so that two variable vector. X is in the n-dimension space. So that to calculate the distance, we calculate the n component differences.
But that would go into the normalization for the difference in x.
Yeah, but I think this large n is only one time appear. So Kangmin, this one was used large n in this paper.
Yeah.
I think. And this large n means it's not the dimension. It's
한치의
a
사이즈인데.
Number of time points. I don't know why.
number of time points.
I don't know why that would scale with the number of time points.
Is it that n?
This is correct. And then if we ignore the large n, we subtract the time series and we inner product, I guess. The dot is inner product.
This is supposed to be like a KL distance. But I don't know if it literally takes that form. In the SNE, this part should be like a KL distance that you minimize.
It corresponds to the KL dimension.
So there you take the log times the difference and then multiply it by.
이거는 5.
근데 어차피 x hat t는 밸류지.
네.
x hat이 벡터였으니까 t 고정하면 값 하나잖아, 그렇지? 값 하나에서 값 하나 빼서 값을 곱한다. 왜 저런 짓을 하지?
Oh, I think on the last part, we should erase head.
The last part.
Y diff head.
So it's just y diff.
Right.
So y diff was
Why would we define y diff hat then?
제곱분의 1 해가지고.
Y diff hat goes in the
근데 y diff hat이랑 y diff 컨셉 나왔을 때 차이 아닌가?
좀 나누기만 하면.
미치겠네.
Yeah. Sorry, yeah. So, the last part is y.
Yes. Okay.
Sorry,
Anyway, the key point is we want to minimize the global factor. Anyway, that's the goal of this algorithm. Global factor means we want to minimize this value.
저 y diff 바가 그 아까 스케일링 팩터가 설명에 타임 에버리지라고 나와요.
Time average.
y diff의 타임 에버리지가 나와요.
So that means y diff was vector,
Yeah.
and then we average of them. That would be y diff r.
Yes.
So that means we want to make this time series average is 1.
average with time.
Yeah, so if time series is 1, 2, 3, then we divide by the 2.
So the normalization is a time average.
Okay, so then actually the goal is very clear here. I don't know still about this 1 over n. Our goal is we want to make difference small, right? And we want to make y is also small if possible. Anyway, goal is we want to minimize this multiplication. Is it good? We want to train the w and z so that the resulting y time series, normalized time series y, normalized time series x are as similar as possible. In particular, we wish this difference is smaller when y is large.
Or y is small.
Difference small maybe makes sense, right? We want to maintain the distance structure between x and y, but why we want to minimize the y as well? Because to me, isn't it more natural just do this? Why we multiply y?
So when the two disagree a lot, then that shouldn't affect the output. So y would be silence when the difference is large.
I see.
No, I don't know, that doesn't make sense either.
If y difference is really small, then we don't care about x difference and y difference differences. 맞잖아. y difference가 매우 작으니까 y 변화가 거의 없을 때는 x difference랑 y difference 차이를 그렇게 신경 안 쓰겠다. 아, 전혀 직관적으로 안 봤는데. 쉽지 않아. It's okay. 미나 없네 우리. 이제 소리.
여기 줌으로 참.
아. 우와. Why we want to minimize? I think as long as we understand that key point, then every other thing will become natural. I don't know, hopefully, and further, actually, there is a negative. Oh man. Hey, come on, there is a negative. So do we maximize or minimize? Can you check? Come on, if we minimize this negative value, that means. Oh,
Also,
but
there's
no absolute value. What's wrong here?
Everything is insanity.
This part is correspond to KL divergence.
KL divergence?
And so
Supposedly, but
uh,
KL is like log of P over Q times Q or
Okay.
so it's sort of like the difference.
Okay. There are KAIST undergraduate, postdoc undergraduate in this room. Seoul National University undergraduate. Competition. Which university is better? 형준, 석환, 재훈. 자, 학교 명예를 걸고 해봐.
the definition the DT is here.
Oh.
And this corresponds to input distribution, output distribution, and output difference. And this corresponds to
very nice. 재밌네. 그러니까 이거부터 가는 게 더 좋겠네. 이게 왜 KL divergence지? Why this is time or KL divergence? Pij minus Qij. So Pij is distance between the I and J point. Qij is distance between I and J point in the low dimension space.
y diff를 곱하는 거 약간 일종의 확률 분포를 곱해주는 거 같고요.
큰일 났다.
I don't think that's the usual form the KL divergence takes, is it?
그 KL divergence 공식이 어떻게 되죠? What is the formula for KL divergence? It's
Yeah, it's like
Q over Q.
log of P over Q
Yeah.
times Q or something like that.
yeah.
Q over Q. 로그.
근데 우리 이렇게 갑자기 시간이 있다가 시간 없는 걸로 가는 이게 왜 이렇게 생긴 거지?
이해가 안 가니?
Most practice we handle about the time series data. Now suddenly they remove all the time and then throw into all this index.
Because I and J in this
왜 이렇게
case is
smart-지?
like time.
이게 사이언스 어드밴스.
The index.
아, 근데 그 성어가 와서는 좀 그렇긴 해. 100% 이해 못해서 그냥 통과해 준 것 같아.
그러니까 이 인풋 데이터 생긴 게 벡터, 투디멘션al 시뮬 클럽.
포스펙 카이스트 긴장하세요. 옆에 지금 재훈이 사픈 것 같아요. 말씀을 너무 힘내.
I'm wrong. So the definition of scale divergence.
Yeah, that is, yeah, K-word divergence. And gradient.
Of scale divergence. This corresponds to.
It's a gradient.
Gradient.
That's not what we're minimizing, that's just the,
Yeah,
it's just
so-
the direction that we change things.
Yeah, sorry, yeah, I misunderstood. The update rule is not to minimize D of T. D of T is the direction.
아, 오케이. 아, 그러면 조금 더.
The absolute value.
아,
Okay, now then it's not the minimize. It was gradient of the KL divergence. They try to calculate the direction then, where to jump and which direction.
Well, that's a big problem for neurons, right? How do they do calculus? So then, that's the Hebbian rule.
제인에 의해서 미분하면 저렇게 된다고 그레이디언트가. 되는 것 같긴 한데.
Right. So our objective is train the W, right?
Yeah.
Then shouldn't we calculate the gradient in terms of W
By using the gradient.
using this gradient?
Using using this gradient. D.
Okay.
But
So with
So we have that is gradient.
gradient, but we need we so while our objective is not change Y, but we need to change W.
but
Yeah,
But the update rule is here. I don't.
So it needs to be the gradient with respect to W.
Yeah.
The update rule is here, but it's too complicated. So
If we know the gradient of y, maybe we can estimate the
yeah.
gradient of w. Isn't it?
Maybe. Yeah.
Maybe from the gradient y, I think we get
Chain rule.
kind of yes. Some
Oh yeah.
C.
That's okay. Then got it.
We need a Mina from data science problem. Or we have one more student from data science group in this room.
Right. But maybe. Anyway, we try to minimize scale divergence between X diff and Y diff. Okay.
I don't know, I know we are very confused about due to detail, but the goal is very clear now. We have two distance structure. One distance structure in the original data set X, which is high dimension. Another distance structure is Y in the low dimension. And we want to minimize their KL divergence. Here KL divergence is whenever we point out 2 points in the X, 2 points in the Y spaces, their distance, their distance, their distance, KL divergence relationships are similar. That's zero. That's our goal. To do it, we wanted to calculate the gradient of KL divergence. To be frank, we don't understand it, unfortunately, but I think best is that KL divergence gradient. Since we know the gradient, then we can use a just gradient descent algorithm thing to go to the small value of the Km. Phew. I don't know, yeah. We can invite this is two Japanese group, right? The other.
Yeah, maybe.
Which university? Can you check?
It can.
We can.
Yeah, it's the same.
Your friends.
No, I have talked to their postdoc.
I see.
Yeah, but,
So which group in the weekend?
we can computational neurobiology.
Okay, gotcha. So one thing is, maybe we can invite him to give a lecture online. So anyway, I look at all the slides. The final results are very exciting. So yeah, unfortunately, we don't fully understand the algorithm itself. Can you move on?
Yes. The algorithm is
Yes.
and from this result. And by using this algorithm,
So three-dimensional input.
yeah, and the entangled data can be separated by using this algorithm. And the linear separability is kind of metrics to evaluate the separation. And the linear separability is very high. And more complex handwritten number data. And PCA doesn't work.
Yeah.
And conventional
Kiss me.
this name method is good. And Hegelian SM and this name is similar form.
So then here's a question. We have Disney already. Why they want to have it? I don't.
Because you want to encode it in biological neurons.
olfactory system. They have
It's for visual system, so it's the same.
Yeah, it's mimics a real biological olfactory system. They think the new method is simpler than
Simpler.
conventional method.
It's not, I don't think this is going to blow the lid off of like a computational, as a new computational algorithm, but in a biological neural network.
So okay. Why I missed the first part? Why they want to do in the olfactory thing? So do they apply this to the real olfactory data set?
They try to similar
Okay.
task. It's the similar chemical components induce similar smell. For example, the linear structure induces a glass-like smell, and this, then ring induce a fruit-like smell. So our olfactory system classify the structure of the chemical structure. So they apply Hegelian to similar task. The input data is chemical structure data, and the output data is mapped to a low-dimensional space. For example, the structure is glass-like smell, and the structure is
So input is what, a sequence or what?
Yeah. It's the input data.
It should not be just picture, right?
いや、なので、あ
High dimension. Object-free data.
I think it's just the firing rate of 50 different kinds of neurons.
Neurons.
I don't know how to.
Maybe there are various bunch of olfactory neuron, which are on or which are off, maybe.
Activity of sensory neurons.
So there are about 20 sensory neurons.
Yeah.
How much they are activated depending on the. Okay, so it's not that bad.
The dimensional reduction that they're proposing doesn't seem mind-blowing. They go from 50 to 32.
Here, about 20 dimension, right? Original is a dimension because we have 20 sensor. So I think this column is a feature.
Yeah.
So 20 dimension and this is all different input.
So 50 to 20.
I think 20 to the 2 dimension or 3 dimension.
Yeah, so 20, and the dimension is this size, vertical and horizontal size. Output data is 2 dimension.
2 dimension, so that all this similar color are.
Yeah, similar.
That's if you only want to classify, what did you say, grass and fruit?
For example.
So this is just a really simple example, like the Drosophila olfactory network is not outputting 2 dimensions.
Yeah, but the result is here. I think it's not clear. The smell is classified to 10 categories. Each color corresponds to the category of smell. I don't think it's not clear, but linear separability is compared to similar
So 50 dimension is going to the 2 dimension.
Yeah, 2 dimension.
And then we wish all these different smells are clustering well. Tiffany looks okay. Why do we need a new method? Still.
I guess this is notorious for is being slow.
Slow? Maybe? I don't know. Tiffany is
It's just
slow.
like
Okay.
people are going to moving to UMAP.
It is
faster.
quite slow, but if you use pair processing, then it's quite usable.
Then why they don't use the UMAP to compare this?
I think UMAP
Better?
is that's my guess.
This is our next paper.
So which year?
Drosophila does
Which
UMAP.
year this work?
Happiest remember the weekend.
Now, another thing that so the Tisna is, in practice, Tisna is notorious for making fake clusters.
Fake clusters.
I'm kind of disappointed that this paper didn't cover
Then another my question is why they always just focusing on 2 dimension?
Visual.
It's for visualization.
Yeah.
The 2 dimension is not a limitation of this method.
Okay. What do you think, Hyun?
I think in 2 dimension, PCA performance of the PCA is definitely downgraded. But if you use the higher dimension,
3 or 4 dimension PC might
then
be okay.
might be better. I think.
I see. What you mean is, if we compare these 3 performance in the 3 or 4 dimension, the difference becomes smaller.
Yes, that's my guess.
Okay. Makes sense.
I think the one thing, I guess one of the guys, some of the guys that have like gene expression data, so single cell RNA data, so you can just, single cell RNA data has like sometimes like a billion cells.
Then
So you can just put in the algorithm and see heavy antisense works best.
maybe you can move forward.
Yeah, there are final conclusions. Again, T3 might be working in the factory section. Yeah, it's different as well. Yeah. And again, T3 consists 3-layer feed-forward network, which mimics all factory circuits in use of life. And they think NPR T3 is simple and can reduce dimension effectively as T3. And the KVR T3 might be working in the old factory circuit in use of it. Yeah, that's all.
Yeah. I know we had a lot of questions. Any other thing to ask?